All right, in chapter 10, we're going to turn our attention from the actual oxidation of food, which we discussed in chapter 9, and how that's used to create cellular energy, ATP. We're going to talk about plants do the process by which they produce food, or this process known as photosynthesis. So the synthesizing part ought to key you off that we're going to be talking about an anabolic reaction, right? We're going to be building something. And anytime you need to build something, you're going to need energy. And in this particular case, the word tells you exactly where the energy comes from, right? Photo, light. The light that the sunlight energy from the sun is what drives the process of photosynthesis, is what allows us to use the laws of thermodynamics, specifically the conversion of one type of energy into another, to survive on the planet. I'll get into the synthesis part in just a second, but I want to talk about light for a couple slides. Light travels to the Earth from the sun in a variety of wavelengths, right? From very, very short wavelengths, like we see down in gamma rays, to very long wavelengths, like we have in radio waves. Now, the wavelength is defined as the distance between two peaks or two valleys. So the gamma rays would be very close together. The radio waves would be very far apart. Now, there's an indirect relationship between wavelength and energy. What I mean by that, if you look at the arrow up above for energy, as the wavelength increases, the energy goes down. So gamma rays have lots of energy, and radio waves have considerably less energy. Now, the other wavelengths of light are broken down, like gamma rays and radio waves, into categories. X-rays are anywhere between wavelengths of a nanometer to 10 nanometers. There's also UV light, infrared. But let's take a look in the middle for a second, what we call the visible light spectrum. The reason why this is called the visible light spectrum, and it's the color of light that we see, we humans see. You notice down below, I'm comparing a dog to a human, so they have a little bit different visible light spectrum. Notice they don't really see greens, they don't see reds. They're similar to us in the blues and the purples, but they're very different in those other colors. All right, now the reason why I bring up the visible light spectrum, we think about plants, right? And I'm talking about not the flowering part of plants, but the part of the plant that actually does the photosynthesis work, the leaves. Leaves are typically green, and there's a reason why that is. The color of objects, whether it's a plant leaf or a stop sign, is actually a reflection of the color that's not being absorbed. So if we just confine our attention to the visible light spectrum, purples and blues and greens and yellows and reds and magentas and all those colors are striking the surface of a plant's leaf. But there is one color wavelength of light that is not being absorbed. It's being reflected, and that's the green. Hence the reason why plants look green. Same reason why stop signs look red. The paint that you paint a stop sign with absorbs all the other wavelengths of light and reflects the red. Now, light, Einstein told us that light travels in two forms. It travels in the wavelength that I've showed you, and it also travels in a little nugget of energy called a photon. All right, it's called the dual nature of light. Some very interesting stuff. But we're going to be looking in the photon more because, of course, this is about the energy transfer from the sun to a plant in order to engage in photosynthesis. The pigments that we're going to be specifically looking at, you might be familiar with some of these, are chlorophyll, right? There are two types of chlorophyll in plants. Now, other plants have other types of pigments, but these are the more common ones. For example, carrots have a lot of carotenoids, which are orange in color. Chlorophyll A and chlorophyll B are similar molecules, just slightly different between them. A has got a little darker color. What you're looking at on the chart here illustrates what I just said. Along the x-axis, along the independent axis are the wavelengths of light, and then along the y-axis is the amount of light that's absorbed by chloroplast, all right? And you notice that there's quite a bit of absorption down here in the blues and the purples, and then there's another bit of absorption that occurs up here in the oranges and the reds, but right here in the greens and the yellows, there's no absorption. So it's not being absorbed, it's being reflected, right? That's, again, the reason why leaves look green. Now, plants absorb wavelengths of light in the shorter wavelengths down the blues and the purples for a reason, but we're going to focus our attention in terms of photosynthesis. We're going to focus our attention more on this upper end, right around 650 to 700 nanometers. Again, we're talking about wavelengths here, very short and very long, relatively speaking. Okay. The interesting thing about chlorophyll molecules is they're made up of atoms, and those atoms are made up of electrons that are susceptible to being excited, right? In other words, they're in a chemical position by which if you give them some energy, like a photon, there's that term, right? So light is in wavelengths and photons, nuggets of energy. You can excite one of those chlorophyll molecules. And when you excite that chlorophyll molecule, specifically what we mean by exciting is you give it energy, right? And so energy is the capacity to do work. So just like we talked about in chapter 9, where we had these energy-rich electrons that we stripped off of food, right? And we could use them to do work. We're using an energy-rich electron here again, but we're just a little different about how the energy came about. The ones in respiration had energy by virtue of their chemical structure in food. Same reason that gasoline has energy, all right? In this particular case, the electron that's being excited the energy is coming from the sunlight, from a photon coming off the sun. Now, where that electron comes from is important because it doesn't come from in the chlorophyll molecule. The chlorophyll molecule actually engages in a reaction by which it takes water. Now, plants need water obviously for hydration, but they also need water for photosynthesis. And in the chlorophyll molecule, what it's going to do is it's going to split water up into its individual components, right? So there's going to be the proton, then its electron, and then, of course, the oxygen. The electron that's being excited is this one right here. So water, in addition to hydration for plants, provides a source of electrons to be excited. Now, once something's been excited in energy, it can fall back down to its ground state. Think of the ground state as being the non-energized state. And when it does, it releases energy again. So kind of like when we were talking back in the chapter where electrons move between shells, electrons in this case can kind of like batteries. They store energy. Now, anytime energy is transferred, this is the second law of thermodynamics, you don't get exactly out what you put in. So, if this is 100% of the photon, you might get the 80% out. There's always a price to be paid to Mother Nature in the form of heat. Heat is a form, as I've mentioned before, of unorganized energy. It's hard to use energy. It's difficult to use heat energy. Okay. What you're looking at on the right too is a flask of chlorophyll that was what they did is they took ultraviolet light and they shined that ultraviolet light onto this chlorophyll in this flask. Then they turned the lights off in the entire room, so it's totally dark. And what's reflected is the energy fluorescing back out again. The reason why it's looking red is that has a lot to do with what it was illuminated with before, which in this case was the ultraviolet light. The point of all this, though, is showing you that chlorophyll molecules do have that ability to absorb energy and then later on release it. So that's going to become important because those molecules being able to give us those energy-rich electrons, it's going to allow us to do some work, right? Okay, let's do a little anatomy, a little form, since I'm all about this form and function thing. Take a look at a typical plant leaf, and if we did a cross-section of your typical plant leaf, this is the top of the leaf, and this is the bottom. At the bottom of the leaf, there's these interesting little areas called stomata, stoma for singular, but a stomata is actually an opening, and those little bean-shaped objects you see around them are known as guard cells. G-U-A-R-D, guard cells. So let me draw you a couple of guard cells. All right, and what's underneath that is the stomata, a hole. Now, what stomatas do is they open and close, and you can see in the picture they let carbon dioxide and water in and out. So when those guard cells shrink back, then the stomata is open. See, they can switch between those two. Now, we'll talk a little more about stomata later on because something very interesting happens when plants open and close their stomata in certain environments. All right, down here in the thylakoid, the chloroplast. Notice it's surrounded by an inner and an outer membrane, very much like we saw in the mitochondria. As a matter of fact, that's another bit of evidence for the fact that chloroplasts might have been their own individual organism at one time because we have a membrane within a membrane. You remember all cellular membranes, no matter what I'm talking about, are made up of these things, right? Those phospholipids. Okay, what's a thylakoid? Each of those little green sacs, they almost look like Mentos to me, is a thylakoid. They're not really individual sacs, even though they kind of look like it. You can see in the picture, they're all connected together. So if you excuse my lack of artistic ability, you get the idea there. A stack of thylakoids is known as a granum, and several granum are known as grana. So you see several grana in this electron microscope picture. The area between the thylakoids, the area between, is known as the stroma. I'm going to use those terms later on, so it's important that you understand how all that's working. All right, so getting back to the light again and putting that in context with the thylakoid, here's what's happening on the membrane of the thylakoid. Remember I said it's all about the membranes. It was all about the membranes in respiration. It's all about the membranes too, or all about the membranes here too. But notice that thylakoid membrane, right? It's reflecting green light or transmitting green light. Either way, it's not absorbing it. It's absorbing the blues, and the purples, a little bit of the yellows, and then the reds and the oranges. So photosynthesis is a set of reactions, and that first set of reactions that we're going to discuss are reactions that occur right on that thylakoid membrane, okay? So let's take our attention and look at that in a little bit more detail. So what's happening right here when light strikes that thylakoid membrane? All right. Before I actually get into the process of what's going on there, let's talk a little bit about the chemical equation, right? So let's go back to the chemistry again for a second, because I want to point out, if you look at this chemical equation, it's relatively or quite familiar to you, right? It's basically the reverse of respiration. Respiration, we took glucose and oxygen, broke it down into carbon dioxide and water, and released energy that we made ATP with, right? Went to the left. Photosynthesis, we take two smaller, less complex, less energy-rich molecules, add the energy from the sun, and turn them into a much richer energy molecule, glucose. And so down below, I've done that little bit of that alphabet soup thing again. You know, like we did in respiration, we said it's, if you don't count the numbers of atoms, respiration is basically nothing more than a transfer of the H. Right? So the hydrogen from the glucose goes onto the water, and in the process, that releases the energy that phosphorylizes, right? Sticks a phosphate onto ATP. Then in photosynthesis, we're just reversing the process. Putting the H back onto the carbon dioxide, and then producing the oxygen, which is given off as the gas, right? The oxygen gas. Now, remember that plants also photosynthesize and respire, so they have mitochondria. Excuse me. Whereas animals, of course, we only respire. We don't photosynthesize. All right, now back to that thylakoid membrane surface that I was talking about. The first set of reactions that are going to occur on the thylakoid membrane are known as the light reactions. And the reason why they're called that is you can see they're driven by light, sunlight. And the light reactions require water. Again, plants require water for a variety of reasons, obviously, you know, hydration. But this particular water is going to be used, as I said, as a source of electrons and protons in the light reactions. We'll see that in a little bit later on. Light reactions give off oxygen. And all of those are occurring on the thylakoid membrane. The other set of reactions is sometimes called the Calvin cycle. Well, it's called the Calvin cycle here. It's also sometimes called the dark reactions. I've also heard these two called the light dependent and the light independent. What we mean by that is the light reactions have to have sunlight. The dark reactions, it doesn't mean they necessarily only happen in the dark. They don't need sunlight. So they're light independent. They happen during the day as well as at night. And notice what's happening in the Calvin cycle. We're taking a simple energy low molecule, turning it into food. We're engaging in an anabolic reaction. Now, in order to engage in an anabolic reaction, you need two things. In order to build something up, you need two things, don't you? You need energy, okay? And you need a source of electrons. And that's where this comes from. Now, that looks similar to something we saw in respiration, right? Looks similar to these guys, those electron carriers. No difference here. I always remember that NADPH is in photosynthesis because it has the P. That's how it reminds me. That's a phosphorus that's on there. But NADPH and its oxidized form, NAD plus, are, again, electron carriers. So the energy comes from now this is the same ATP that we make in respiration. It's just that this particular ATP is going to be used to do something in the Calvin cycle, whereas the ATP we talked about in respiration is used to do, pretty much anything. So we have our two components for building a molecule, right? For engaging in an anabolic reaction. We have the electrons that we can use to reduce things from the NADPH, and we have the energy by which to do that from the ATP. And as those electrons are used and as that energy is used up, then as you can see in the top, ADP and NAD plus, the oxidized form, are going to be cycled back to the light reactions. All right, now we're going to look at the light reactions in a little more detail, but this is a good slide to kind of organize your thoughts. I've already talked about most of this. The only thing I didn't mention to you is that the Calvin cycle happens in the stroma. All right, so, right there, that's good. Oh, stop. All right, so the light reactions are on the thylakoid, the Calvin cycle, the dark reactions are in the stroma. All right, let's go into the light reactions now. The light reactions require light. So here we are on the thylakoid membrane. There's your phospholipids, right? Very familiar with that. And embedded in cellular membranes, remember, there are proteins, right? And do all kinds of things. This particular protein embedded in a thylakoid membrane is known as a photosystem protein, all right? And that photosystem protein is sensitive to wavelengths of light. And those wavelengths of light carry packets of energy known as photons. And we're going to eventually use that energy in the photon to excite an electron. Let me remind you once again that source of electrons is water, right? Hydrogen ions plus their electrons plus oxygen. So that's the electron that's being excited. All right, now all those little lily pad pigment molecules, pigment is color, right? Those are all pigment molecules, and what they act like if you were to take a hand lens out onto the sidewalk and focus it on a piece of paper. Paper doesn't burn at normal atmospheric temperature, no matter how warm it gets here in the summertime. Paper doesn't burn until it gets 451 degrees Fahrenheit. That's the reason Ray Bradbury named his book that. But what happens with these lily pad pigment molecules, you notice the arrows bouncing around? What they're doing is essentially focusing the energy from the sun onto a spot. So while paper won't burst into flames at 90 degrees, if you focus the sun's rays using that lens, you can get that temperature up to 451 degrees, indeed get the paper to burn. Okay? So that's what's happening here. All of that energy from those different pigment molecules, it's not just one pigment molecule absorbing a photon, they're all absorbing them, right? And they're jumping from lily pad to lily pad essentially until all of that energy is getting focused onto a special pair of chlorophyll molecules found in a reaction center. And that's where the business occurs. Right? In the reaction centers where the electron from water is going to be excited, energy is going to be given to it, and it's eventually going to be passed to an electron acceptor. Okay, now, that's what goes on. Where are we at this point? At this particular point, we have an energy-rich electron. Right? At this particular point, we have a means by which to do work. I'm pausing there because I want you to think about the fact that you have heard this before. We talked about it in respiration. Now, I keep going back and forth between respiration and photosynthesis, not because I'm trying to confuse you. I'm trying to unconfuse you, because so many times on exams, students get the two processes mixed up because they are very similar. So make sure when you're studying this, you're looking at the similarities and the differences, right? Okay, so now we got an excited electron. What can we do with an excited electron? We can do some work. So let's transfer our picture. Here's the photosystem we were just talking about. All right, I'll explain in just a second why it's called photosystem 2. All right, here's the sunlight energy. There's the water. There's the source of electrons. There's the oxygen that's going to be given off. All right, now the electron's going to be excited just like we talked. So we were right here when we left the previous slide. want to step back for a second, though. When you split up the water molecule, where do the protons go? Well, they get dumped inside the thylakoid. All right, so you're going to end up, excuse me, in a situation in which the thylakoid has a very high proton or hydrogen ion concentration. It's actually going to be higher than the stroma. Lots here. Not too many here. That's familiar, isn't it? Okay, now back to the electron. The electron's got energy. What are we going to do with that? Well, we could move things uphill with energy. We could turn on a pump. So the PQ and the cytochrome complex together act as a pump that pumps protons from the stroma into the thylakoid, thereby adding more to that disequilibrium. Sound familiar, doesn't it? Same thing that happened in the mitochondria, right? All right, here's where it becomes a little bit different. In the mitochondria, when we were done with that well, for one thing, there's only one pump here, and there were 3 in the mitochondria. But once that electron gets through those pumps in the mitochondria, its energy was spent. And that's where oxygen came in. Remember, oxygen came in at the very end of the process and accepted that electron. That's what allows the whole process to work. Remember, electrons can't move along until the one in front of it essentially moves. There's got to be things called electron holes. So oxygen has to take the electrons away from the mitochondria so that your NAD and FAD can keep dumping more in. So it's a similar kind of thing here. So that electron, as it leaves that cytochrome complex, another particle that's going to help move it along, passes it to another photosystem. And I'll talk in just a second why that's called photosystem 1, even though it seems like that should be numbered the other way around. It's not a mistake in your book. In that second photosystem, we're going to excite that same electron off of water, same electron off the hydrogens of water. We're going to excite it again. A little bit different wavelengths. Photosystem 2 on the left has a reaction center that we call a P680. That means it's very sensitive to wavelengths of light of 680 nanometers. Photosystem 1, the reaction center is called a P700. A little bit longer wavelengths of light excite that one. All right, so we're going to excite that electron a second time. Now, that is different than in the mitochondria. We're going to pass it to a helper particle. So I'm following the yellow arrows, folks. And eventually, that electron is going to find itself in the company of an enzyme, NADP plus reductase. Enzyme, right? Ends in ASE. Sometimes the name of an enzyme also tells you what it does, doesn't it? NADP plus reduces, or I'm sorry, NADP plus reductase reduces NADP plus. It puts electrons on it. What electrons? The ones that were stripped off of water, the ones that were stripped off of the hydrogens in the water, the ones that we excited, the ones that we used to turn this pump on, the ones we excited again, that's the same electrons that we're going to add to NADP plus and a proton to make NADPH. Remember, we're in the light reactions here. There were 2 products of the light reactions that we needed from the next set of reactions. One set of reaction turns another, and one of them was a source of materials, and there they are. You remember when we talked about enzymes, though, going back to this idea of exciting it again? I said that enzymes don't eliminate a cell's need for energy. It just allows a cell to carry on processes, cellular process, with less energy, not zero energy. So the energy, as well as the material, the electron itself, is what drives this process by which that enzyme NADP plus reductase creates NADPH. So we're making a simple, more complex